The OctoChrome FISH assay simultaneously examines all 24 human chromosomes in a single hybridization on one slide. First, prepare metaphase spreads on the specific 8-square slide. Then, perform an automated scan of the sample slide to facilitate location of all the metaphases and future re-evaluation of all cell images. Next, position the 8-square slide over the OctoChrome device and hybridize the probes. Results obtained can show 3 different painted whole chromosomes in each square based on the arrangement of chromosome combinations on the OctoChrome device.
I first had the idea for this AlterChrome FISH method when I started to apply FISH in human population studies during the early 1990s. Then I introduced my idea of triple color painting on an 8-square slide to Cytocell at the annual AACR conference in 1994.
The main advantage of this technique over existing methods like classic chromosome binding and modern spectral karyotyping is that it can readily analyze all 24 human chromosomes in a larger number of cells.
Begin with the preparation of metaphase cells fixed in suspension. As a control, pipette a drop of cells onto a slide and verify appropriate density. Now, dip an 8-square OctoChrome slide into pure ethanol for 2 minutes. In a sequence of alternating squares, distribute 5 microliters of cell suspension onto the template slide. To prevent the cell spreads from interfering with each other, air dry the samples before spotting adjacent pairs. Apply 20 microliters of 0.2 micrograms per